Today we are going to learn basically respiration, but before diving into its further details, it is very essential for us to understand metabolic pathways. So first we should comprehend what metabolic pathways are. Basically, metabolic pathways are the intricate series of chemical reactions that basically um, control life within our cells. These pathways are essential for maintaining life uh, and could be broadly classified into two main types. You see in the slide, one is catabolic and another one is anabolic pathways. Now question is, what are the catabolic pathways? Basically, catabolic pathway involve the breakdown of complex molecule into simpler ones, releasing energy in the process. The primary goal of catabolic pathway is to extract energy stored in large molecules such as carbohydrate, fats and proteins, and then convert it into usable form, usually in the form of ATP. So, in this series, one interesting example which you are going to learn in this series of lecture, that is cellular respiration, basically which involves breakdown of glucose to produce, or you can say to generate, ATP molecule. And then you see that in addition to this catabolic pathway, there are certain pathways there is another pathway known as anabolic pathway. Basically, this sort of pathway involve the synthesis of complex molecule from simpler ones, requiring an input of energy, whereas catabolic pathways release energy. So the main function of anabolic pathway is to build and maintain cellular structures and molecules. And these anabolic pathways generally use energy stored in ATP to drive the synthesis of macromolecules like proteins, nucleic acid and cell membranes. We would like to share some examples of anabolic processes like protein synthesis, DNA replication and the synthesis of complex carbohydrate, that is glycogenesis. And these anabolic pathways are basically very much essential for growth, repair and maintenance of cellular functions. So, therefore, you see that catabolic pathways break down large molecules to release energy, whereas anabolic pathways build complex molecules consuming energy in the process. So, taking together, these two types of pathways basically contribute to the overall energy and maintenance of cellular structures and functions in different living organisms. So, you see that now today we are going to learn the process of respiration and how cellular respiration proceeds. So, you see that cellular respiration is a vital process that occurs within the cells of living organisms to generate energy. Basically, it involves the oxidation of complex organic molecules such as glucose through a series of enzymatic reactions. These reactions occur in the presence of oxygen, that is aerobic respiration, or without oxygen, known as anaerobic respiration. So, the process of cellular respiration can be just summarized into main categories like glycolysis, oxidative decarboxylation of pyruvate, citric acid cycle, electron transport chain and oxidative phosphorylation. Now, let us begin with the glycolysis, which is the initial stage of cellular respiration, and this process takes place in the cytosol of the cell. So, the seat of glycolysis is basically cytosol. So, we are going to just further details of this glycolysis. So, you see that in glycolysis the raw materials are basically glucose molecules, which are 6 carbon sugars. And the main objective is to convert one glucose molecule into 2 molecules of pyruvate, which is a 3 carbon compound. And the main motive of this breakdown of glucose is, of course, to extract energy in the form of ATP to carry out various cellular functions. Now, what is meant by glycolysis? Question arises, what is meant by glycolysis? Basically, the word glycolysis originates from the combination of two Greek terms you see, that is, well, glykis means sweet, and lysis means breakdown or splitting. And this glycolysis is also referred as Emmeden-Meyerhof-Parnas pathway, or abbreviated as EMP pathway. So, before going into the details, we would just like to have a sketch of this whole process. So, basically, glycolysis is a metabolic pathway consisting of 10 steps in which generally glucose molecule undergoes conversion into 3 carbon pyruvate molecules, and simultaneously 2 ATP molecules are generated, and 2 molecules of NAD are reduced to NADH. This pathway occurs aerobically as well as anaerobically. As you know that aerobic respiration is a metabolic process that occurs in the presence of oxygen, while anaerobic respiration is a process that occurs in the absence of oxygen. This is quite clear. Now, generally, lactate is formed from pyruvate as part of the glycolytic process, and the sequence of reactions in glycolysis varies among species, primarily in the regulation mechanism and the subsequent metabolic fate of the produced pyruvate. So, in aerobic organisms, generally what happens, glycolysis serves as the precursor to both citric acid cycle and electron transport chain. Basically, it functions as the central pathway for catabolism of glucose. Now, this is just a whole sketch of the complete glycolysis process, and then we can go through in detail step by step what this pathway or how these pathways are performed and how these are regulated. We will discuss in the coming part of my lecture. So, basically, glycolysis initiates the breakdown of 6 carbon glucose molecule into 3 carbon pyruvate molecules, and the enzymatic reactions involved could be divided into 2 distinct phases. One is preparatory phase or phase 1, or another one is payoff phase. So, what is preparatory phase? So, basically, the process of glycolysis involves a series of 5 steps where glucose undergoes enzymatic phosphorylation using ATP, resulting in the formation of fructose 1,6 bisphosphate. Subsequently, this fructose bisphosphate is broken down into 3 carbon compound, namely glyceraldehyde, 3-phosphate and dihydroxyacetone phosphate. This initial phase of glycolysis leads to the cleavage of hexose chain. To facilitate this cleavage and prepare glucose for conversion into 3 carbon compounds, there is a requirement of how much ATP molecules? 2 molecules, as you could say, as initial investment in this process. So, you see this diagram shows this slide shows the steps involved in preparatory phase, as just we have outlined it. Now, payoff phase. So, basically, this stage represents the final 5 reactions in glycolysis, where ATP molecules are generated as glyceraldehyde 3-phosphate and is then converted into 2 molecules of pyruvate. During this process, 4 molecules of ADP are phosphorylated to form ATP. Despite the formation of 4 ATP molecules, the overall outcome is only 2 ATP molecules per molecules of glucose oxidized, and 2 ATP molecules are consumed in basically phase 1. Now this this slide shows the steps involved in payoff phase as just we have outlined. Now how the process of glycolysis proceeds, we will go into the details of these steps and how each step is catalyzed. That is very interesting to share and discuss. So the step first is phosphorylation. Although you see that glucose serves as source of energy, it must undergo a process to elevate its energy level before entering into the glycolysis. So basically this elevation is achieved through the phosphorylation of glucose molecule. And the initial step of glycolytic pathway involves the phosphorylation of glucose or at its 6 carbon, resulting in the formation of glucose 6-phosphate. And basically this reaction is facilitated by enzyme hexokinase, and that transfers a phosphate group from ATP to substrate. Since phosphorylating glucose is energetically unfavorable, it requires coupling with an exergonic reactions involving hydrolysis of ATP. In majority of the animals, plants and microbial cells, hexokinase is responsible for phosphorylating glucose molecule. In this reaction, the presence of magnesium ion is very much essential as substrate for hexokinase reaction is basically manganese, and the role of manganese is very crucial as it helps or you can just shield the negative charges of ATP, making the terminal phosphorus atom more susceptible to nucleophilic attack by an hydroxy OH group of the glucose. Now second step is isomerization step. Generally in the second stage you see that transformation of glucose 6-phosphate through isomerization takes place, and basically the carbonyl oxygen originally located at C1 is shifted to C2, resulting in the conversion of aldose glucose 6-phosphate into ketose known as fructose 6-phosphate. So basically here the main motive is conversion of glucose 6-phosphate to fructose 6-phosphate, and the enzyme responsible you see that is for catalyzing this particular step is referred to as phosphohexoisomerase or phosphoglucoisomerase or glucose phosphate isomerase. This enzyme plays a very crucial role in converting the pyranose ring of the glucose into furanose ring of glucose. Moreover, you see that isomerization is very important for two main reasons in glycolysis. First, when the phosphorylation happens at carbon 1, it is easier to add a phosphate to a simple primary hydroxyl than to a hemiacetal group hydroxyl group of glucose 6-phosphate. Second, during the following steps where a carbon-carbon bond is generally broken, the isomerization of fructose with the carbonyl group shifting to carbon 2 activates carbon 3, making the cleavage of carbon-carbon bond easier. So this is the rationale of this particular isomerization. Now in step third, that is you see that in the preceding step, the carbonyl group of glucose 6-phosphate shifts to carbon 1 from carbon 1 to carbon 2, leading to the availability of hydroxyl group at carbon 1. The enzyme phosphofructokinase basically catalyzes the phosphorylation of this hydroxyl group and more or less in the similar manner like first reaction, ATP provides the phosphoryl group, and the process basically requires the presence of further magnesium ion. The phosphorylation of fructose 6-phosphate is an basically endergonic reaction and serves as another priming step. The conversion of moreover, you see that the conversion of fructose 6-phosphate to fructose 1,6 bisphosphate facilitated by the enzyme phosphofructokinase, which is a crucial point in utilization of glucose for energy production rather than its diversion towards other sugar pathway, or could be for simply storage purpose. Therefore, the reaction governed by PFK1 phosphofructokinase stands a very crucial regulatory checkpoint within the glycolysis and could act as a sort of switch to regulate glycolysis. Now, step 4 of glycolysis, you see that fructose 6 bisphosphate aldolase breaks down fructose 1,6 bisphosphate into 2 triose phosphate by splitting between its carbon 3 and carbon 4, and this process produces dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Although the reaction can proceed in both directions, you could say it reversible, it predominantly favors the formation of fructose 1,6 bisphosphate. The equilibrium of the reaction leading to the formation of 2 triose phosphates from 1 fructose 1,6 bisphosphate is generally influenced by concentration of all the 3 compounds involved, including the substrate and the resulting products. It is very important to note at this point. The enzyme called aldolase, due to its resemblance to the reverse process of an aldol condensation reaction, and is strongly endergonic. Aldolase is basically an enzyme protein composed of 4 subunits, that is a tetrameric, and could serve as an example of covalent catalysis. This enzyme basically initiates the reaction by combining the keto group, keto carbon at position 2 with alpha amino group of lysine residue located in the active site of the enzyme. Now, step 5 is you see that the enzyme triose phosphate isomerase, or you can say TPI, triose phosphate isomerase, basically facilitate the conversion between the 2 triose phosphates generated by the preceding reaction, namely 3-phosphoglyceraldehyde 3-phosphate and dihydroxyacetone phosphate. As glyceraldehyde 3-phosphate, you see that is a key participant in the primary step of glycolysis and serve as a substrate in the subsequent glycolytic reactions. There is a tendency for an increased conversion of dihydroxyacetone phosphate into glyceraldehyde 3-phosphate to maintain the equilibrium in the process. Under a standard condition, the reaction is considered endergonic. However, a low intracellular concentration of glyceraldehyde 3-phosphate causes a shift to the right, allowing both you see that triose sugars produced from glucose molecules to progress it further into glycolytic pathway. Consequently, carbon 1, carbon 2, carbon 3 of the initial molecules become equivalent to carbon 6, carbon 5 and carbon 4 respectively. So, the triose phosphate isomerization is basically reaction marks the completion of the initial phase of glycolysis, In this process, each glucose molecules undergoes glycolysis of glyceraldehyde 3-phosphate with the consumption of 2 ATP molecules up to this point. The next phase of glycolysis involves the payoff phase of glycolysis. That is very interesting to discuss here. So, payoff phase starts basically from the conversion of aldehyde into carboxylic acid. So, basically this part of the glycolytic pathway encompass the partial oxidation of triose phosphate along with the series of reactions responsible for converting the terminal phosphate group of phosphorylated sugars into ATP. But you see in the slide, the initial step basically in glycolysis involves the oxidation of glyceraldehyde 3-phosphate, and the conversion of glyceraldehyde to its acid form is generally linked to the reduction of NAD to NADH. The energy released during this oxidation process is preserved by forming a carboxylic acid anhydride bond with a phosphate group commonly referred to as an acyl phosphate to form 1,3-bisphosphoglycerate catalyzed by the enzyme glyceraldehyde 3-phosphate dehydrogenase. You see this you see that glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate by the enzyme glyceraldehyde 3-phosphate dehydrogenase. So, in this particular step, due to you see that high potential for energy transfer, 1,3-bisphosphoglycerate exhibits a strong inclination to transfer its acyl phosphate group to ADP molecule. So, basically this results in the synthesis of ATP, and 3-phosphoglycerate is formed by a process catalyzed by phosphoglycerate kinase. So, basically this step is very crucial, and this is the first substrate level phosphorylation reaction, which is very crucial for generating ATP molecule. So, now question arises that why only certain molecules, certain compounds are able to transfer its phosphoryl group to ATP molecule. So, question should come in the mind that why only certain compounds they are able to donate its phosphate group to ADP molecule, why not other intermediates they donate its phosphate group, because simultaneously there are number of intermediates they are present inside the cytosol, but only certain molecules, certain compounds which they are able to donate its phosphate group to ADP molecule. So, this could be answered that there are certain substrate which are having more phosphoryl group transfer potential than ATP molecule and other compounds present in the cytosol. That's why these compounds they are able to transfer its phosphate group to the ADP molecule to synthesize ATP molecule because of the higher substrate phosphoryl group transfer potential than other compounds. So, this basically substrate level phosphorylation reaction is crucial for generating ATP, and each glucose molecule yields 2 triose phosphates. 2 ATP molecules are produced through this mechanism, and the phosphoglycerate kinase reaction basically serves to offset the energy debt incurred in the priming reactions. And this step is also very much facilitated by magnesium and to transfer the phosphoryl group to the ADP molecule. ADP as the true nucleotide substrate, ATP serves as a true nucleotide substrate for this particular reaction, and considering the production of 1,3-bisphosphoglycerate as an intermediate in the 6th reaction, the 6th and 7th reactions of the glycolysis are appropriately viewed as coupled reaction. Now you can see how 1,3-bis phosphoglycerate is converted into 3-phosphoglycerate, and then 2 ATP molecules are synthesized by the phosphate group transferred from the substrate. So this is very interesting example of substrate level phosphorylation, where substrate is donating its phosphate group to ADP molecule because of having higher phosphoryl group transfer potential than other intermediates present in the cytosol. Now you see that this substrate 1,3-bis phosphoglycerate has high phosphoryl group transfer potential than ATP molecules, and rest of other intermediates who could not donate its phosphate group, they are having lower phosphoryl group transfer potential than ATP molecules. And one interesting thing I would like to share at this juncture, that phosphoryl group transfer potential of ADP lies somewhere in the mid than the other intermediates. Now next step of glycolysis, that is phosphoglycerate mutase. This enzyme basically facilitates the movement of phosphate group from 3rd position to the 2nd position in 3-phosphoglycerate and result in the formation of 2-phosphoglycerate molecule. And at this step, the presence of magnesium is very crucial for this endergonic process. Initially, the enzyme phosphoglycerate mutase is basically activated by 2,3-bis phosphoglycerate to phosphorylate histidine molecule located at the active site, forming phosphohistidine. In the course of the reaction, the phosphate group is transferred from phosphohistidine to the substrate 3-phosphoglycerate, creating an intermediate 2,3-bis phosphoglycerate. Subsequently, the enzyme-bound intermediate undergoes breakdown, restoring the phosphorylated histidine of the enzyme and releasing the product, that is 2-phosphoglycerate. At this stage, the presence of magnesium is very much crucial for the reaction to take place. Notably, this enzyme is absent in chloroplast, preventing interference with the Kelvin cycle where 3-phosphoglycerate is basically produced. If phosphoglycerate mutase were present, it could have divert carbon from the Kelvin cycle to glycolysis. That's why this enzyme is absent in chloroplast. So this is very interesting to share with you. Now you see that next step is catalyzed by an enzyme enolase, which basically facilitate the transformation of 3-phosphoglycerate into phosphoenol pyruvate, characterized by its high potential for transferring phosphoryl group. So basically what happens that enolase reaction converts a substrate with a comparatively low free energy of hydrolysis into product, that is phosphoenol pyruvate, which generally possess a significantly higher free energy of hydrolysis. And it is very important to note here that both 3-phosphoglycerate and phosphoenol pyruvate have a similar potential metabolic energy content when considering their breakdown into inorganic phosphate, carbon dioxide and H2O. Basically, enolase plays a crucial role in restructuring 2-phosphoglycerate by eliminating a water molecule, transforming it into a state and that can release more potential energy through the process of hydrolysis. And the enzyme is basically hindered by fluoride ions in the presence of phosphate. Now you see that how this reaction is catalyzed by enolase. 2-phosphoglycerate is basically converted into phosphoenol pyruvate, and this step is catalyzed by enzyme enolase. Now phosphoenol pyruvate basically produced in the previous reaction have a very high phosphoryl group transfer potential. This is very interesting to note that one compound I mentioned 1,3-bisphosphoglycerate, and now this phosphoenol pyruvate is having higher phosphoryl group transfer potential than ATP molecule. So basically this reaction, enzymatic reaction, is facilitated by enzyme pyruvate kinase, and this is again a very interesting example of substrate level phosphorylation. And basically this reaction involves the transfer of a phosphoryl group from phosphoenol pyruvate to ADP molecule, and in this reaction also magnesium ions are necessary for this reaction, and its activity is basically enhanced by the potassium ions also. The significant alteration in the free energy during the conversion of phosphoenol pyruvate to pyruvate primarily arise from the high favorable and spontaneous shift of pyruvate from its enol tautomer to the more stable keto form following the phosphoryl transfer. Moreover, half of the energy released from the phosphoenol pyruvate hydrolysis is preserved in the ATP phosphoanhydride bond, while the remaining energy drives the reaction forward. So this is the sketch of the reaction, how phosphoenol pyruvate is converted into pyruvate. So this is again a very interesting example of substrate level phosphorylation in the process of glycolysis. Now how for each molecule of glycolysis metabolized through glycolysis, 2 molecules are consumed during initial 5 reactions which constitute the preparatory phase of glycolysis, and during the payoff phase there is a generation of 2 ATPs and 2 NADH, and you see that one glucose molecule produces 2 triose phosphate, and each triose phosphate produces 2 ATP and 1 NADH. As a result, there is a net gain of 2 ATP and 2 NADH in whole process of glycolysis. However, the number of ATP molecules could vary depending upon the kind of carbohydrates metabolized and also the process of hydrolysis involved in sucrose or starch. So this is the just overall net gain during the process of glycolysis. So thank you very much for patient listening my lecture. In my next lecture we will discuss how different switches of the glycolysis are controlled, what are the control points of the glycolysis, and how the different intermediates serves as a regulator for the controlling the process of glycolysis. Thank you very much.